When I got my diagnosis of breast cancer, it felt like the loneliest moment of my entire life. Because I had cancer. And there was nobody that could change that fact.
It is a very lonely thing the moment you hear it. The next thought after the wow, this is happening to me, can often be, am I going to die?
I think the hardest part was the month's delay between the PET scan and the biopsy. And of course you're thinking, well, this tumor's growing and I've no idea if it's growing at a crazy rate or what's happening.
I could see and feel that fear on them. As a mom, you're trying to protect your kids, and you can't protect your kids from this. And it's so helpless feeling.
Hearing that you have a disease like cancer is as devastating today as it was 20, 30, or 50 years ago. Diagnoses from Alzheimer's to cardiac disease continue to devastate millions of patients and families a year, despite decades of research in traditional medicine. Precision medicine could change that. Across the healthcare and life sciences industry, there is enormous excitement about what precision medicine can do, and enormous concerns that its power will be drained away by systems that can't support the science. And the only way we can make precision medicine work is to come together. A good place to start is the patient journey, where we all have new roles to play in screening, diagnosis, and treatment, if we want to realize the incredible outcomes that precision medicine can deliver.
Precision medicine was born really out of a new way of diagnosing people. When you think about the history of medicine, a patient would come in, a physician would examine that patient, and for the most part, it was outside of the body that we were trying to diagnose disease. Then came the advent of all of our imaging technologies, and we were able to see inside the body. We were able to look for broken bones. We were able to look for different things that were going on inside the body in a big way. Then came the genomics revolution. And when we started to really push and look at the DNA associated with each tumor inside a human, that opened up the door for new ways of diagnosing patients and working through a diagnosis in a very well-informed and holistic way.
Genomics was becoming integrated in every discipline in research. And I was actually being trained during my postdoctoral fellowship in functional genomics and systems biology. I applied those skills to the question of differential biology, just comparing tumors from different groups of patients. And as I started to see patterns of genes behaving differently in the tumors of women who had pretty much the same diagnoses, I was really interested to investigate whether there were genetic reasons for that, whether genetic ancestry, for instance, the shared genetic ancestry of African Americans and West African women could be driving biological differences in the tumors. Honestly, at that time, my theory of there being differences in tumors across patient groups really wasn't considered a legitimate hypothesis.
Researchers like Dr. Davis have helped prove that biological markers, known as biomarkers, influence everything from whether someone develops a disease, to how it progresses, to how well treatments work. Biomarkers are medical indicators that we can measure and track. Blood pressure and cholesterol levels are biomarkers. So is blood sugar. With precision medicine, there are now many biomarkers that we can detect, monitor, and use to develop more targeted treatments for different patients with the same disease. And the bank of known biomarkers is always expanding.
The field of biomarker research has exploded over the past 10 years. We have more targeted therapies that are being approved at higher rates than we ever have before. And the reason that is the case is because we're understanding more about the basic biology of tumors. What makes a tumor initiate and grow and metastasize?
I have grown up in this field in the last 25 years and seen us go from having no therapies for certain genetic conditions or for certain cancers to having choices. I have seen us go from having a handful to maybe a few dozen different genetic tests that could really help change the trajectory of someone's healthcare to hundreds, if not thousands.
The word that gets thrown around a lot is actionability. Some clinicians will say the only actionable biomarkers that exist out there are those that have a therapy tied to them. Maybe that's not the case. Maybe there's no treatment today, but can they be enrolled in a clinical trial? That should constitute actionability. But we don't have a consistent language to talk about all the different ways in which biomarkers can be beneficial to a patient. So we need to get agreement, at least on areas like that.
The idea of precision medicine, for me, is to use that information to understand, based on what's actually going on in that individual's tumor, rather than a large cohort of patients who we all assume have the same mechanisms of disease, to identify the actual mechanisms of disease.
And the scientists at the bench, as they learn more about that, the drug developers are able to develop very precise molecules that target those specific mechanisms underlying tumor biology. And it's really that nexus of those two things coming together that has brought about this sort of revolution in cancer therapeutics that we're calling precision medicines.
In traditional medicine, treatments are developed based on average responses in a large population. In precision medicine, patients are stratified into smaller populations based on genetic variations, disease risks, and response to treatment. Because of her family history, Shireen Carter makes annual mammograms a top priority, except in 2018. Balancing a successful career in pharmaceutical data science with a busy personal life, Shireen put off her exam by 6 months. Her story shows why one-size-fits-all screening tools aren't the right fit for everyone.
The type of breast cancer that I was diagnosed with is called triple negative. Unfortunately, it's very aggressive. And also, unfortunately, it's missed in between mammograms. So I asked my surgeon, when do you think this started? And my surgeon said, given the size and how fast this grows, 6 weeks ago. Which means that if I had done everything the way that I always did, went in February, had a mammogram, I probably wouldn't have gone back until the following February. And I may have missed having cancer at stage 2, and I probably would have been at stage 3, if not stage 4. That delay was probably, in fact, what saved my life and gave me a chance at getting the right treatment.
Mammogram screening has its limitations, but it's still a key factor in the earlier diagnosis and more productive outcomes we see in breast cancer versus lung cancer. Until recently, we've had no lung cancer screening tools. Well, that left a patient like Richard Gray relying on chance, or an observant radiologist performing a routine neurology MRI.
The MRI turned out to be good from the neurology point of view, but the results came back saying there's an abnormality in your lung that you might want to get looked at. Stage 1. So, yeah, it was a small, I think about 1.5 centimetre tumour, something like that. Well, I said to Dr. Auchincloss, if they hadn't by chance found this during this unrelated test, what would have happened? And he said, well, you'd have probably come to see me in 2 years' time with stage 4 cancer, and I probably wouldn't have been able to do very much. So, yeah, dodged a bullet there.
Breast cancer, we have great screening processes, and they continue to get a little better with the use of MRI and other high-resolution scans. Lung cancer, we're just starting. And so we're trying to figure out who we need to screen and how often. At least we finally have a test that's approved, but much more work needs to be done. We have to keep moving forward in trying to study our population. Everybody thinks smoking is the number one cause of lung cancer. And what we're learning is that the majority quit smoking. And now we're seeing a growing number of people who never smoked, who are now being diagnosed. And those are a different biological population.
My smoking history is 0. I've never smoked. My mum and dad smoked when they were younger. They stopped when my mum found out she was pregnant. So I've never really been around smokers, and I've never smoked myself.
One of our biggest dilemmas down the road will be, how do we screen for people who have never smoked but are still at risk of developing lung cancer? Science and precision medicine is going to have to help us dictate what those risk factors are and who should be screened.
Not every cancer is a cancer that is ideal for screening. Sometimes we want to know where we can test patients that have the symptoms that make them more likely to have the cancer. Because then instead of testing 300 million Americans, we might be able to test 2 million. And if we can test 2 million people to find 100,000 cancers, that's a much more economic test paradigm to follow. So screening doesn't need to be testing everybody walking down the street to see if they have cancer. That could lead to false positives or false negatives.
One way to improve screening today is to avoid testing large numbers of people for a single cancer that very few are likely to have. In the future, we could be screened for many cancers in simpler, less expensive ways.
The key to curing cancer, or at least making it a chronic disease, is to find it very early. I really believe that one day, not in my lifetime, but probably my kids, where you will go to the doctor and you will have your height and weight taken and your cholesterol measured, but they will also do a blood draw and they will be able to test very quickly whether you have cancer or not. That's what we call liquid biopsy. We will use it, I think, not only to screen, we will use it to diagnose, and we'll also use it to monitor. I think it's going to be a game changer in how we treat this disease.
Because of precision medicine, health screening is becoming more targeted, more accurate, less invasive, and less costly. Being able to make better decisions about who to screen for what diseases is one small example of the power of data in precision medicine.
Data is key to help us rationally use precision medicine. You can have the technology to develop a test, but if you don't have the data to show where it is useful, it's not a useful test. So I think in the realm of data, we are racing to keep up with the technology. One example of this is liquid biopsy. Recently, there's been one test approved for testing whether a person is at increased risk for recurrence. It's called minimal residual disease, whether there might be cancer cells or cancer DNA circulating in that person's bloodstream. But at this point, the data is not there for what we do to sort of reduce that risk. This is one example of where we really need to develop the data to help guide physicians treating their patients and help guide patients with kind of what their care needs to be moving forward.
Within precision medicine, data is king. And in fact, when you get outside of oncology, it gets even more complicated when we start to talk about neurology-based or infectious disease-based precision medicine. getting inside the patient with different pieces of data that we get both from lab tests, from overall looking at the patient, from taking a piece of tumor outside of their body, and looking at all of those different types of things and leading all of that data into one complete picture for the patient allows us to better pinpoint exactly what to do with that patient and bring it forward into a diagnosis, a treatment, and potentially a cure.
Having lost her mother to breast cancer, Terry Conneran began mammograms early and was vigilant about her health. But when she started having trouble catching her breath, she blamed things like summer humidity or dust from home renovation projects. Her primary care doctor thought it might be allergies or asthma. But the pills and inhalers didn't help.
A couple days before Christmas, my husband's like, "Why'd you go to the doctor? Like, you've been feeling lousy for quite some time. Get your antibiotic and move on." I went to my PCP, and she could hear a lot of fluid in my lungs. It justified doing a chest x-ray. The chest x-ray highlighted a tumor that was the size of a small grapefruit in my lung. That's not how it got diagnosed. That's how we found that there was a problem. Because a chest x-ray is not a great way to see something. I'm back first of the year. I'm not feeling any better. In fact, I'm feeling kind of like worse. She puts me through a number of different tests, pulmonologists, CT scans. And over the course of around 3 weeks, I went in for a biopsy. And the doctor called me. And he said, "You have a non-small cell adenocarcinoma."
So much has changed over the last several decades. We now are able to take a sample from a patient even before they've been diagnosed with cancer, a small biopsy through a needle, even a blood sample, a sample from urine or other body fluids, and be able to interrogate that sample to determine the likelihood that the patient actually might have cancer. And what that allows doctors to do and surgeons is take the patient in for surgery with the knowledge of what the likelihood is that that patient might have cancer. We can also test patients today before they go to surgery to, with a high degree of confidence, rule out cancer so that they can avoid having that unnecessary, costly, and invasive procedure altogether. So we've just made incredible progress, and I think we haven't arrived yet. I think the progress will continue.
As diagnostic advances in precision medicine evolve, they could reduce the incredible toll that waiting for answers takes on patients and their families, and allow life-saving treatments to start sooner. "2007 was an exciting time in Kelly Brook's career. She made partner and was focused on work. And that's where she was when she felt a lump in her breast, so concerning, she went to her doctor right away. But because she'd had a clear mammogram only a few months before, her doctor said it was probably a cyst and might go away on its own. Not willing to wait for answers, she insisted on a needle aspiration. She was ultimately diagnosed with breast cancer. Kelly was put on the same aggressive course of treatment as all other patients with her diagnosis at the time.
When I was diagnosed, it was a bit more binary. The treatment that I got was based on the size of my tumor, the fact that it had spread to my sentinel node, and that it was estrogen positive. So I was going to have these things, and everybody that was in my category had those 3 things, and that's the way it was handled. If your tumor was over a certain size or if under a certain size but it had spread to your lymph nodes, then you needed to have chemo. If it was small enough and hadn't spread, then you just had radiation. There was a treatment, and you got slotted into what kind of cancer you had, and then everybody that was in that tranche had that kind of treatment. I think one of the big side effects for the kind of chemo that I was having, the most noticeable side effect is the loss of your hair. And as soon as my doctor told me that that was going to happen, I said, well, maybe it won't happen to me. I have thick hair, so maybe I won't lose my hair. And she just kept saying, you're going to lose your hair. And that was probably one of the hardest messages, other than the diagnosis of cancer, obviously, that I received, because you look in the mirror and you look like a cancer patient when your hair is gone. So that was tough for me. And it wasn't too terribly long after I had finished my treatment, so maybe 2 or 3 years later, studies came out that said, oh, now we probably don't need to give that kind of chemotherapy to everybody, because that's overkill.
How we choose medications or drugs to treat a person's cancer has changed a lot over the years, especially with precision medicine. We've been able to really tease out different types of cancers and determine really what are the appropriate therapies, what are the effective therapies for a patient, characterizing the cancer based on different protein expressions, and subsequently to that, gene expression has enabled us to more effectively treat cancers and personalize treatment for patients based on the type of breast cancer they have. The majority of breast cancers are hormonally driven. We call them hormone receptor positive and HER2 negative. And for these patients, many of their cancers are primarily driven by estrogen. And so the efficacy of chemotherapy can be limited or none for some of these patients. And with clinically available molecular tests, we are able to determine for a patient whether this person is going to benefit from chemotherapy. And in many cases, the answer to that question is no.
I was participating in a clinical trial. The way the trial was designed, they added another agent to the standard regimen. And that agent is in a class of drugs known as platinum drugs. The platinum drug is not something that a triple-negative breast cancer patient would get at that time if they weren't in a clinical trial. It wasn't until after I found out about my mutation, I read that in Europe, individuals who were diagnosed with triple-negative breast cancer and have the same mutation that I had, they are given a platinum agent that actually probably had quite more of an effect on my particular cancer because of my genetic mutation than if I wasn't in the clinical trial at all. I wouldn't have known all of that, and my care team certainly wouldn't have known all of that, because from their perspective, getting genetic testing wasn't going to change their treatment plan. What that showed me was how critical getting tested is, even if at the very beginning there are no decisions that are going to be made. There are still decisions that could be made after the fact. Like, do you want to remove any organs? Do you want to have a different type of surgery? What is your risk later on in life, not only for the cancer that you presently have, but in case there might be others that come up? My plan is to get tested probably in another 5 years or so, because every decade, the bank of information on genetic mutations can change. Scientists will not only learn more about the genetic mutations that they're familiar with, but they also may find new genetic mutations that they hadn't realized previously.
We have to still emphasize to everybody out there is that when or if you are diagnosed and they're ordering all the MRIs and the PET scans, everything, and getting a tissue diagnosis, what is equally important, and I consider incomplete diagnoses, is that you must have your genomic biomarkers performed. But it changes things dramatically if we find one of the 9 or 10 markers that are FDA approved for lung cancer. And it gives the patient an opportunity to receive a smart therapy, a pill, or a precision medicine therapy, as opposed to just trying with chemotherapy. To watch what the field of lung cancer has done has been quite revolutionary. In the early 2000s, as a clinician, we felt very helpless with patients, only being able to offer them chemotherapy that we knew wasn't going to be that effective and probably was not going to help their quality of life that well. I could have brought in someone who was a trainee, a fellow, and they could have picked any one of these drugs, these 2 drugs on a piece of paper. It was a matter of which side effects did you think the patient could tolerate based on the drug type. I didn't really feel like a specialist. What kind of special decision was I making if anybody could choose these therapies? Fast forward 10 years and the whole thing changed. Now I feel like a specialist because you need to have that knowledge of what biomarkers match with what therapies. We now could be smart, and we had to be thoughtful about what our choices were as clinicians, and we could think more now about the patient as an individual and what was unique about their particular cancer that they were fighting. And the good news is we've had multiple therapies in the same family. That's epidermal growth factor receptor, EGFR. If you have this gene mutated, we are up to the 4th different therapy already, the 4th generation of drug. And each generation has improved on top of it. And what precision medicine has allowed us to do is to look at them as individuals, figure out what's unique, and now we're able to move these therapies to earlier stages.
I'm now on a drug, an EGFR inhibitor, that is normally used for stage 4 patients, even though I'm a stage 1 patient. I've had some people express a little bit of surprise that I'm getting it with a much earlier stage tumor than would normally be prescribed. That's something where a genetic test proved that this medication would be good for me. Dr. Auchincloss said he thinks he got all of the tumor out, but there may be a few remaining cancer cells, and this medication should help kill those.
Traditional cancer chemotherapies, they target the hallmark of cancer, which is a rapidly growing and dividing cell. And so the earliest cancer chemotherapies were really great at targeting DNA and rapidly dividing cells and killing those cells specifically. What they also did was target any other cell that is rapidly dividing. And so those are things like hair follicles or the lining of your intestines, and that's why you have the very broad side effects of hair loss, GI issues, neuropathy.
Many early on were scared to leave their home because they feared the side effects of the cytotoxic therapy that would occur. Now, I will add that even our most toxic chemotherapies, which we don't have to use as often anymore, we've seen improvements as well. So even when we don't find the appropriate genomic target, we can actually use a combination of immunotherapy and chemotherapy, which is highly effective, which has less side effects than even the 2-drug therapies we had in the early 2000s. And patients are having a lot of success with this.
In the age of precision medicine, we're taking advantage of these molecular mechanisms that are governing cancer, that are specific to the tumor. So these would not be happening in any of the normal cells in our bodies, but only in the tumor tissue itself. And so the hypothesis was that we would be able to develop much more specific, much more efficacious drugs that had a much more tolerable side effect profile. And that has been the case. And in fact, we can administer these drugs in the outpatient setting. Folks don't need to be hospitalized. They can take a pill bottle home and don't have to be monitored as they take them.
And now we encourage people to take vacations. We'll give them a month's supply and go. You're okay. Do it. It's really emancipated the patient from having to feel like they have to stay close to their doctor, stay close to their hospital. And gosh, go take that cruise, for God's sakes. Go play golf across the seas. No, now the biggest side effects that we counsel people on, especially with these pills, is acne, mild diarrhea, and not getting too much sun exposure, because some of them can make you more photosensitive. And that sounds like some antibiotics, really. So don't just plan the one cruise or one vacation, plan it for the next couple.
I initially was not sure whether to tell people because I didn't want, lots of sympathy, and I didn't want everyone to think, oh, goodness, he's going to be dead in 6 months. I talked about it with my wife, and we initially thought, let's keep it quiet. But then we thought, why not? Why not talk about it? Why not? Why keep it in? And so we did tell everybody. Actually, that worked out well. People were not avoiding me and pretending, trying to change the subject. Everybody was very supportive when I was going in for the testing and the surgery. And we decided to turn it into a positive thing and start up a way of funding our surgeon to do more work. So we had a gala in our garden in June and raised $30,000 to continue his work, and hopefully it's something we'll do every year now to celebrate the fact I was cured.
We can diagnose someone, we can do it earlier and do it more effectively, and figure out what's in their tumor to then give them the appropriate therapy, the oral therapy by mouth, for years afterwards. But people are no longer being defined by their disease anymore. They are defining themselves.
But at the end of the day, it really comes down to that DNA that is in my body, that's in your body, and how much we can learn about it to make sure that the best, most customized drug, and in my case, it was a platinum agent, will actually have the greatest effect. By having the right treatment for the genetic mutation that I had, all the cancer was gone, and thankfully I could go into surgery and subsequent radiation more as a preventative from having the cancer come back, as opposed to still trying to get the cancer out of my body.
What moved me to be a breast cancer specialist is both the personal relationships you develop with patients and kind of walking beside them as they go through their cancer journey, as well as the hope that each day brings in terms of research breakthroughs that improve incrementally, but improve cancer care every day. Through the technology we have available to us, we can help patients either be cured from their cancer or live with their cancer in a way that it does not affect their longevity or their quality of life. Precision medicine has helped achieve that through helping us access and accelerate various technologies, including immune therapy. Some patients have been fortunate enough to have what I'll call dormant cancer for, the foreseeable future.
One of the ideas we had for the gala that we did was that everybody could pick up a bracelet when they came into the gala, and there were 3 bracelets. I have one for survivor, there was another one for family of a cancer sufferer, and then one in memory of an amazing person who'd passed away as a result of cancer. One point I made was that almost all of us are going to wear one of those bracelets, and probably a good number of people are going to wear all 3. And that's sad, because really you should be wearing none of them, ideally. If you don't wear a survivor of leprosy bracelet, or my my father got the plague, that's been fixed, and smallpox has gone away. And so we should do that with cancer. If medicine's going to do its job, then we should be aiming for no bracelets.
Precision medicine works. We as a collective have developed the most precise and targeted therapies that have ever been invented, and patients are getting them and receiving benefit. It works. What we're doing now is optimizing how that whole system works, democratizing that access to every patient who is diagnosed with cancer. And that's where we talk about these barriers to access and removing those barriers.
The science is ahead of the healthcare system's ability to deliver the science. There's a senior executive in the pharmaceutical industry who back in 2016, she spoke about, in this country we have Star Wars medicines and a Flintstones healthcare system. And I think that is a telling comment from a senior executive in the healthcare system, that we have this innovation, we have these incredible therapies that can cure disease, but we can't necessarily always get them to the right patients.
Our current healthcare system is based on traditional medicine, with limited numbers of tests and treatments for a given disease. Precision medicine requires us to develop and select from growing numbers of targeted tests and treatments for smaller patient populations. As a result, we're experiencing what's known as leakage, or gaps in the system, where precision medicine doesn't reach the patients who need it. There are many causes of leakage, and they're all complex.
The big ones that I see in our day-to-day work is education. Making sure that the patient is informed and knows that these testing modalities and targeted therapies are available to them and are alternatives to the traditional chemotherapies, and that they should have that conversation with their doctor. And then educating the clinician so that when a patient does bring that up in the exam room, that they're armed with the information to be able to have that intelligent and informed conversation about whether or not a targeted therapy or a precision medicine approach is appropriate for that patient. And that's where we spend, and I personally spend the majority of my time, is closing that education gap with the providers themselves. And it's not because these are inexperienced, unmotivated, or uneducated clinicians. It's not that. Rather, it's just new medicine. And because it's new medicine, we've got to fill those education gaps and provide them with the scientific backstop so that they can understand fully what tests they're ordering, the data that comes back from those tests, and how to connect that information to the pharmacopoeia that they have at their disposal. We serve a network of community-based medical oncologists. This is where 80 to 85% of all oncology care is taking place. It's not happening in your large academic medical centers. It is happening out in the community.
If you're at a place like Memorial Sloan Kettering or MD Anderson, where everything is internal, you tend to see very little leakage. The systems are specifically set up and all the work is done in-house. But when you're in the community setting and everything goes out and you don't have the funding or knowledge to be able to create bridges between these facilities to facilitate the process of getting material from where it is to where it needs to be, that's where you see the biggest amount of leakage. That's also where there's the least amount of knowledge, and the more somebody could really benefit from an expert interpretation of these complicated test results, they're much less likely to have access to experts. In my experience, having run precision medicine programs in community oncology settings, there's a whole host of reasons why there's leakage that is often not typically being measured. Sometimes it's as simple as filling out a paper requisition form or an online requisition form, not in an automated way directly from an EMR, such that there's a mistake made in that, either from a coding perspective or misspelling the patient's name, so that even if everything works correctly after that point, that report may never come back into that patient's chart to then be utilized.
I should also mention that there are very practical barriers to participation here. Things like tissue availability. Is a disease biopsyable at all, right? Because in order to get this sort of molecular information, you've got to have some sort of tissue to do the analysis on. Now, thanks to advancements in technology, we're able to do blood-based assays now and other sorts of biological specimens, and that has helped tremendously in democratizing access to these technologies. But tissue is the gold standard today, and tissue remains the issue for many of these tumor types where it's very difficult to get a good biopsy that you can send off to a laboratory.
I remember at one point, I was managing a molecular diagnostics laboratory doing next-generation sequencing, and I had a very large oncology hospital that sent me a lot of work, and they started having a lot of failed samples, and they were very upset that all their samples were, a certain percentage of samples were failing a much higher than anticipated rate. And they complained, and they said, "Why is this happening?" I looked into it, and I realized that the samples that failed were always coming in on the same day. They were always being accessioned in pathology on a Monday morning. And what was happening was those samples were coming out of the OR on Friday afternoon and not being properly fixed. They were sitting through the weekend. The DNA is degrading, and the DNA is necessary for these tests to work, obviously. And so giving that information back to the pathology groups that managed that particular hospital got them to change their procedures to ensure that all those samples coming out Friday afternoon were still being properly fixed, which prevented these errors in the future. In the community oncology setting, where you have pathology groups that have agreements with local hospitals to perform certain services for them, may have an adverse incentive to not send those specimens quickly or not get reimbursed for handling part of this process and delay that process for a variety of reasons that may negatively impact the turnaround times. And then that results in an oncologist having to make a decision sometimes without this information. So when the information comes, it's too late and they don't find value in performing that service. So then they're less likely in the future to order that service again, or it was an expensive process. Other things could be payers not paying for these services, so patients being stuck with bills, complain to the oncologist. Now the oncologist doesn't want to incur an unnecessary cost on that patient, and so now is less likely to order those tests again in the future.
We talk about the cost of cancer care and the impact on health plans, but really what we want to do is avoid waste. We want the right treatment to go to the right member at the right time. And that's really where the opportunity is in precision medicine, is to fine-tune who is most likely to respond to a given therapy. And if that person isn't likely to respond, then maybe there's another therapy or what's considered a standard therapy that would be their best option. And that's what we want.
The science of precision medicine can only be as powerful as the ecosystem that supports it. And within that ecosystem, we need collaboration between everyone, from doctors, researchers, drug developers, and diagnostics companies, to pathologists, payers, and of course, patients.
Will we get there? We'll absolutely get there. But we can only really get there by deconstructing the system as it is today to identify those roadblocks that prevent these things, precision medicine, from actually working as it can, as well as it can.
Within precision medicine, there is vast potential and enormous obstacles. And in between, there are people.
Because I happen to be seated in the Ivy halls of academia, and I get to talk to these scientists every day and see the data, I want everyone to have the kind of information that I have to benefit from the technology that I'm using.
We can exist in this ecosystem together in a collegial way, knowing that we still put the patient at the center of what our goals and objectives are.
You know, we're all just regular people trying to do almost the same job, improve the outcomes for members or patients.
We feel very connected to our patients. Their successes we take as our successes, and their challenges we also take as our challenges.
I hope we do get to a point where precision medicine isn't some lofty thing that only those with financial access or those who live in certain parts of the country, but that it's felt by everyone, because everyone deserves the chance to live.